Hi, my name is Jimmy Yonts, and this summer I participate in undergraduate research at the University of Michigan in the field of environmental engineering. I work with Professor Raskin and graduate student Nadine Kautlars on a project investigating bacterial communities in Ann Arbor drinking water.
So, you may be surprised to find out that even though our water has been filtered and disinfected down at the drinking water treatment plant and meets US EPA regulations, it can still contain hundreds of millions of bacterial cells per liter. And we are exposed to these bacteria every single day through drinking, showering, and various other activities. And for most individuals, these bacteria present are perfectly harmless, but some of the bacteria can cause infections in those who are immunocompromised. And we call these bacteria opportunistic pathogens.
Our primary research question is, why do opportunistic pathogens grow in tap water and household pipes? This is an important question to ask because if we can understand why these bacteria are growing, we can design interventions to reduce their growth and protect public health.
Today, I'll be running a staining procedure utilizing the chemical compound DAPI. I will walk through this procedure while also providing a little more insight into the sort of research I do here at the University of Michigan. The general procedure we follow can be divided up into 4 distinct steps: sampling, filtering, staining, and counting. To filter, we bring the samples to the lab and filter 15 milliliters of the sample we just took through a 0.2 micrometer filter. This pore size is smaller than nearly all bacteria, so we can be confident that the bacteria present on the filter is essentially all of the bacteria that was present in our water sample. The next step involves slicing up the filters and staining them using the chemical compound I mentioned earlier known as DAPI. What DAPI does is complex, or bind, with any DNA present in the sample. DAPI is also capable of passing through intact cell walls, so it can stain both live and dead cells. Once it is bound with DNA, it is capable of fluorescing under ultraviolet light. These characteristics make it a very useful tool for determining the total number of cells in the water. Once the filter pieces have been stained and mounted on slides, we can look at them under the microscope at 1,000 times magnification. Under the microscope, each cell will fluoresce and appear blue, allowing us to be able to count how many cells there are per unit area. We then apply a scaling factor that accounts for the volume of water used and the total surface area filtered. Doing this allows us to accurately measure the concentration of cells in the tap water. DAPI staining has given us a better understanding of what may be influencing the growth of bacteria in household pipes.
DAPI staining is just one step along the way on our march to better understanding why opportunistic pathogens grow in household pipes. I've enjoyed my time so far and look forward to continuing my work on this project in the future. I also look forward to applying the knowledge and experience I've gained from working with Nadine and Professor Raskin when working on any new projects in the future. I hope you enjoyed this short video, and thanks for watching.